So welcome everyone. Today we have with us MD Fayad Hasan. He is now working as a senior scientist in Vala Sciences, San Diego, California. So I'd like to introduce some of his extra curriculum expertise that Dr. Fayad Hasan has. So he is because I closely was a close friend like that, of course, as a close friend, as a close colleague. So he is actually is a good artist, one of the expert artists. You will get some of his outstanding digital art in his paper. Also, he is a very good chef. He invented so many delicious recipes by his own. And now today you'll find his outstanding research work. So welcome, Fayad Hasan. Please start your presentation. Thank you.
Thanks, Dr. Wayne. And thanks for inviting me for today's journal club. So I hope you can see my presentation. So today I wanted to present a little snippet of my research, and my recent research is on neurological disorder in a dish. So what I try to do in general is to mimic a human brain inside a laboratory dish. So what's the motivation between that? So neurological disorders each year affects like millions and millions of people all around the world. And my focus was two of the main neurological disorders, which is epilepsy and Alzheimer. So our brain is actually composed, as you all know, that are composed of like billions and billions of neurons who like interconnect with each other intricately. And because of this, the brain needs a really perfect and really healthy environment. If anything bad goes into the brain, or the interconnection between these neurons are somehow affected, what happens is that our brain cannot function properly, and we start to forget. That can be the Alzheimer's disease, and we can start to convulse or go into seizure, which is the epilepsy. So, but the problem with these neurological disorders is that unlike other diseases like fever or inflammatory disease, for most of the cases, we don't know how this disease actually happened. What's the pathophysiology of this disease? So how do you develop a drug for a disease for which you don't know the etiology? If it's not a genetic disease, some of the epilepsy and Alzheimer are genetic, but most of them are just sporadic and happens at a random stage of the life. So how do you come up with a drug for these things? So the best way would be to get like millions of people, lots of people like this, and give everyone disease. And then what you do, you give everyone a separate drug, and then see which one survives or which one's disease is cured. So then you can say, okay, so that's the cure of the disease. So that's the medicine we have found. And this process is really expensive, so much expensive that a single medicine or single drug, it takes 12 to 15 years to make, and it costs more than $1 billion. And it involves like thousands, like 10,000s of compound screening, and then 200, on average, 250 of them goes into preclinical development. On average, only 5 of them goes into clinical development, and out of this 10,000, we finally can get one drug as in the market. But most of these compounds, so they fail. 80% of them fail in this drug discovery and preclinical developmental stage. And it's the main cause is improper recapitulation of disease phenotype, which translates that the system you're testing these compounds that is not perfect. In simple terms, like I was saying, that you get like millions of people and give them a disease and then give them a drug. So you cannot get people and then give them disease. It's illegal as of now. So the thing we give the diseases to are not perfect. So mainly animal models are not perfect. So what happens is that animals are not human. So in a lots of research, what happens is that you give these animals some compounds and expect that the animals will behave in the same way the human do. But in a lot of cases that doesn't happen, and that results in a failure in the clinical trial, and that the whole thing now becomes like really expensive. To work around this problem, what people are doing now and recent advent of technology is that a stem cell technology came in. So in stem cell technology, what happens is that in a simple way, you get a patient and you do skin biopsy on his hand. And what this means that you get like a small piece of skin from them. And by doing genetical engineering in this skin, you can actually get heart cells, which is cardiomyocytes, neural cells, or kidney cells, or any kinds of cells. So, and now what you have is that you can get cells from patients, not from animals. So, and these stem cell derived neurons or heart cells, these cells, so they give like a good chances of getting like actual results, actual human-like results from these compounds. But with these stem cells, what people started to do is that get the stem cells and make neurons out of them and then culture them on this just petri dishes on a flat surface. So it kind of look like this. But our brain doesn't look like this. Our brain's parenchyma is a three-dimensional object. And in there, the neurons are like positioned in a three-dimensional space. And it's not also only neurons. There are like multiple other kinds of cells also in our brains, like astrocytes and microglia, these kinds of cells. So neurons are the main cells in our brain which communicates to each other and enables us to think. But astrocytes and microglia, these kind of cells, they support their function, and they are also like really crucial for the whole function of the brain and also for the diseases. So because of that, what new technologies have allowed us to make three-dimensional brain-like cultures or brain-like spheroids, organoids, or 3D cultures in a dish. So these three-dimensional objects, so they mimic more like brain. So they even have brain-like structures, brain-like cellular compositions, and they can be made from stem cell-derived neurons, astrocytes, and microglia. So which means that these are like 3D small brain-like things that originated from a human patient who has a disease. And they now are promising to give us a better understanding and better view of what happens in a human brain with a disease like Alzheimer or like epilepsy. So with this background, so what I was trying to do during my PhD is that to make a epilepsy model in a dish. So epilepsy was my main focus at that time. And when I first got into this research work, I found that the main problem with epilepsy in a dish is that epilepsy, if it's not a genetical epilepsy, what happens is that these cultures don't have epileptic seizure-like activities normally. You have to put PTZ or some other chemicals to make them actually make these epileptic seizure-like bursts. And what I'm saying about saying this because if you put like lots of electrodes on an epilepsy patient's head and you record their neurons or the electrical activity, you will see that a patient with epilepsy will have a lot of bursts or synchronized activity in their brain when there is a seizure. So, and that does not translate in these cultures. To solve this, during my PhD, I developed these three-dimensional cultures. So these three-dimensional cultures, I'm not going into the development or what's the process, but it's a 1 millimeter culture, and it's three-dimensional, and it was made from human cells. So when we made this culture, we saw that these cultures kinds of look like this. They are 1 millimeter in diameter and 100 micrometer high, and they have a 3D structure and also have laminar brain-like structures in them. And it kind of looks like this. And we stained them with neuronal and astrocytic marker. So it's just different kinds of colors to put to see which cells are which. And we saw that this culture spontaneously made this nice red laminar structure of neurons, and also this green astrocytic layer, which we also see in the brain, if you see in the brain cortical layers. And this is just a 3D reconstruction of the same thing. And this is the epilepsy recording I was telling about. Like if you put a lot of electrodes, you will see that the brain is firing a lot at the same time. So that's called epilepsy. So to see if we can find this epilepsy inside our culture, what we did, we infected them with optogenetic marker. So one interesting thing about these neurons, that these neurons talk to each other, and when they talk to each other, so their calcium level increases. So if you put this optogenetic marker inside the neurons, what happens is that when the neurons talk to each other and the calcium level increases, they glow. So you can actually see them talking to each other. With this thing, then we recorded these cultures, and we found something like this. If the video plays, you can see that when we added these cultures, and this is a real-time video, and we saw that there are like spontaneous big bursts of neuronal activity, and they're all synchronized, and which looks a lot like epileptic seizure. If you just put a region of interest around it and plot the brightness, you can actually see that you can actually plot these things. So let me play this one more time. So yeah, you can see that there is a clear bursting activities, which looks a lot like epileptic seizures. So if you plot them, it kind of looks like this. We also did electrical recording from these cultures, and it also showed the same thing. So indeed, it proves that these are actually electrical neuronal activities. And we previously we showed that there are like a lot of spheroid and organoid culture, and we also made those kinds of culture, and we showed that our cultures, these red ones, have longer burst activities than those spheroid and two-dimensional cultures, which means that our cultures are much more better at mimicking the epileptic seizure-like activity in dish. So why do we use it? So then what we did was we screened different types of epileptic epileptic drugs on them, and we saw that these drugs actually work on these cultures. So here you can see that when there are no drugs, the cultures are like bursting like crazy, and when you add more and more drugs at a physiological concentration, the burst decreases and eventually goes away. So which shows that these cultures actually can be used as a disease model for epilepsy, which like in plain language, our plan is to get a lot of these kinds of cultures and put a lot of different kind of compounds and see which compounds works to get new and novel and improved epileptic drugs. So you might now ask that if we already have an epileptic drug for epilepsy, why would we need to make more? So this is like one kind of drug, and it works on some cases of epilepsy, but a lot of kind of epilepsy is drug resistant. And for this, with that kind of epilepsy, there are like no cure or no drugs. So we can use this model from the patients with drug resistant epilepsy to get new drugs for them. So then we found out that, okay, so we made these 3D cultures. But one interesting thing is that these 3D cultures are made in this 1 mm confinement. And when we make this 3D culture in there, we saw that we always get this kind of shape. So we changed the confinement shape. When we made it smaller, we saw that the shape actually changed again. We made it more smaller and it became like kind of a blob. So that gave us an interesting perspective that we can also play with the shape of this culture. So to play more with it, we actually made these long elongated cultures. And here we actually saw that in these cultures, at the center of the culture, the neurites are kind of aligned. So this is important because in our brain, the neurites are normally aligned, and neurite alignment is a big topic of research. So we also showed that that can happen. And then we provided a mathematical model that can describe all of these things. That can show here that these green ones are the neurons and red ones are the astrocytes. And previously we were showing that when we put these cells together, what happens is the astrocyte goes towards the outer side and neurons gets to the inner side, and then the neurons clustered to each other. So mathematically, we also showed that how that happens. So this is important because like with this model, you can calculate that in a brain when there is a surgery and like you excise some part of the brain and then the cavity of the brain, so that actually can increase or also can decrease. So you can kind of predict that how the cavity's shape will react with this model, and that can help physicians to decide the next clinical steps. So we also tried this model with our previous cultures, and these black ones are actual cultures and the green ones are simulated cultures. And as you can see, for each cases, our mathematical model predicted that how the cultures would look like. So after this, we also added astrocytes to these cultures. So as I was saying, astrocyte and microglia are like two other important cell lines, and we actually saw that astrocyte actually increases these cultures activities. So why we did that is that we wanted to play with it, and our main thing was to mimic the brain on a dish, right? So we want to see play with all the aspects which can change this neuronal interconnection, so that we can fine-tune and make a culture that behaves just like the way we want it to. So with that thinking, so we started with another planning. So there are like two main kinds of neurons in cortex, human brain cortex, which is one is glutamatergic neurons and one is GABAergic neurons. In a just simple way, what happens that if you have like four neurons who are like talking to each other and they're all glutamatergic neurons, so these neurons have a voltage. So this black line say that's a voltage. So when one neuron fires, the one neuron talks to this neuron. So the voltage of this neuron increases a little bit. If this one talks, it increases further. If this one talks, then it increases further. But if the voltage increases above this dashed line, what happens is that that neuron actually fires, and that excites the next neuron. But if one neuron is GABAergic neurons, so these GABAergic neurons are like people who like talk less. You can think about it like that way. So then if one is GABAergic neurons, then if the glutamatergic neuron talks, so the voltage increases. If this one talks, increases again. But when the GABAergic neuron talks, the voltage actually decreases, and this neuron cannot fire. So with that in mind, we made also cultures with different proportions of glutamatergic and GABAergic neurons. And we saw that when there is like all glutamatergic neurons who just positively increases voltage of other neurons. So then we saw that this curve is a cumulative probability function of burst duration, which means that these cultures are firing a lot. But when we add a little bit of GABA neuron, which is 25% GABA neuron and 75% gluta neuron, we saw that the activity actually decreases. And we add more GABA neuron, the activity actually decreases a lot. So here you can also see the same data. So with this, what we now want to do is that get like different kinds of ratios of astrocytes, glutaminergic and GABAergic neurons to get just the right proportion of things so that this cultures is not bursting. So because like with astrocytes and GABA neurons, you can change the culture's activity, right? So if you make a culture that's not bursting, and then if you get use the same proportion to make a culture from a patient with genetic epilepsy or just basic epilepsy who has like a genetic signature. So if we make it with the patient's cell, so our thinking is that now probably we'll have some seizures, and then we can use that culture to make a drug for that patient. So right now I am working with a Stanford professor, Dr. LaRocca, to make a model of juvenile epilepsy. So he got this like lots of juvenile epilepsy patients who doesn't have any cure, and like these kids are like really not in a good shape. So we're trying to make epileptic drugs for them. So after epilepsy, we're thinking to go more into these cultures and use it more. So because of that, we started working with Alzheimer's disease, and that was my journey at Mayo. So in Alzheimer's disease, what happens is that in your brain, there are a lot of plaques or like bad things that accumulates. And because of that, your neurons starts dying. And here you can see the left one is a healthy brain, and the right one is the like severe, probably postmortem Alzheimer's disease. So in Alzheimer's disease, the neuron dies so much that your brain actually shrinks. But this Alzheimer's disease, like people, lots of companies are working with lots of techniques that how to solve this problem. So obviously the first thing was that, okay, so your brain have this deposited plaques or deposited bad things. So if we can remove them, maybe that can solve Alzheimer's disease, but that is not happening. Even if you remove the plaque, the disease does not go away, and people starts to forget more and more, and eventually gets into a really bad condition. But another thing that people are kind of, it's less explored, is that like these new neurons in our brain kinds of looks like this. So this left one is a neuron and right one is a neuron. So the neuron has this long process with that it talks to this other neuron. So if you look close inside this black box here, that's an axon. So you will see that there are like lots of microtubules inside that axon. And these mitochondria, so they are like running in that axons. So if you now zoom into this mitochondria, so these lines, so they are the microtubules inside the cells. So if you now zoom into these microtubules, you will kind of see that here the mitochondria like move along each other. And in Alzheimer's disease, or in short AD, what happens is that there is a protein called tau, which gets hyperphosphorylated, or it just changes a little bit and becomes these red balls. And that actually breaks down these microtubules. And these mitochondria actually moves on these microtubules using these motor proteins. So these purple and green ones. And they use that to like just like a car or like a jeep on a highway. It just moves around it. And if the highway is broken, they cannot go on different directions. But why is that important? That's because, so this is the actual mitochondria on the left in our lab. So it was imaged with electron microscopy, and right one is the like graphical representation. And what happens in this mitochondria is that you probably have heard that it's a powerhouse of cells. So this mitochondria actually has a motor inside it, which is called the ATP synthase. So this motor actually rotates, and when it rotates, what happens is that it produces ATP. And with this is called oxidative phosphorylation, or OXPHOS. By using this, what happens is mitochondria generates power we eat food, and this mitochondria uses that food to generate powers. So this power is needed for the neurons to communicate or talk to each other, which is really important. So because of that, we have now a hypothesis that the mitochondria in brain mitochondria, when the Alzheimer's disease happens, what happens is that mitochondria cannot move. And because of that, this mitochondria cannot go to this synapses. So these synapses are like these parts, where at the point where two neurons meet each other and talk to each other. So if mitochondria cannot go to these synapses, then these neurons cannot talk to each other properly, because their talking needs some energy or power. So this you can kind of say that these neurons become too lethargic, or they are too hungry, so hungry that they cannot talk to each other. So to prove this, like we have to first prove this, like if this is the case or not. So our hypothesis is based on the fact that in post-mortem Alzheimer's disease brain, these sites, like you will see that there are almost no mitochondria at that site. So to prove this, and to prove our highly convoluted hypothesis, which I'm not going too much into now, we've made neurons and astrocytes and made these kinds of cultures. And in these kinds of cultures, what happened is that we got cells from healthy people, and we also got cells from Alzheimer's disease patients. And what happened is that we saw here that in this Alzheimer's disease patient, there is actually a more phosphorylated tau. So the thicker the line is, that means the protein is more here. So you can see that these load patients have more phosphorylated tau and less acetylated tubulin. So which means that the tau, I was talking that tau gets hyperphosphorylated, and because of that, the microtubule breaks. So acetylated tubulin means the stability of the microtubule. So it is decreased. That means the microtubules are now broken, and phosphorylated tau is increased. That means there is a hyperphosphorylated tau. And Ph.D. 95 is a synaptic protein marker. That's also decreased. That means that these neurons is actually not able to talk to each other properly. And these are like mitochondrial proteins. And you can see that in all cases, the mitochondrial protein are decreased in neuron astrocytes from Alzheimer's disease patients. So which actually adds merit to our claim that, okay, so there is a mitochondrial problem inside the Alzheimer's disease brain. But then we found that there is a drug which is called CP2. So this drug we're working on. So we tried this drug on multiple animals with Alzheimer's disease, multiple mouse with animal models of Alzheimer's disease. So in our lab, we have this mouse who are genetically engineered to have this Alzheimer's disease-like phenotypes. So these mice, when we fed them this CP2 or these drugs, what happens is that all of their Alzheimer's disease-related bad phenotypes went away and they were healthy. But as I was saying, that proving a drug works in animal is not enough. We have to prove that in human system. So that's why we're doing this patient-derived cell culture and trying our drugs on them. So this is the structure of the CP2 of the drug. So we found that CP2 actually protects again oxidative stress also in our neuron astrocyte co-cultures. From the green one is the healthy person control, and red one is the Alzheimer's disease patients cultures. So this at an initial level shows that, okay, so this CP2 is not actually toxic. It actually protects these neurons and astrocytes from oxidative stress related injury. So then we also looked into the mitochondrial proteins in these cultures with the application of MCI or our drug. And we saw that when we add our drug, the mitochondrial protein actually increases. in each cases. So that means that in Alzheimer's disease, the mitochondria were unhealthy, and we are showing that, okay, so in human neurons, in astrocyte co-culture, if you put these drugs, then the mitochondrial health actually increases a lot. So now, so this is an interesting figure. So this is actually a figure showing a neuron and its processes and its stain for mitochondria. So now we have to prove that, okay, so actually the mitochondria is moving. Is that thing affected in Alzheimer's disease or not? So, but that thing is like hard to analyze, because as you can see at the center of the picture, the cell bodies, in the cell bodies, there are like so many mitochondria that you cannot see anything. But you can, if you look here, you can kind of see individual mitochondria. So here we can actually visualize how the mitochondria are moving. To solve this problem, now I developed this new microfluidic devices where they're like one 3D culture here, the culture I was showing, and these are like small microchannels. And here, if you image it here, you can actually see that these are like individual mitochondria. And at the bottom, you can actually see the mitochondria is moving, and at the very bottom, you can see the same video, but using AI, we now can specifically detect which mitochondria are moving at which way, and you can like detect all of them in a high throughput manner. So right now I'm developing this for this assay system in a like 384 well plate, so that you can calculate and analyze like 384 cultures or 384 compounds at the same time with these mitochondrial movements. So with this system, we saw that, yes, actually in the Alzheimer's disease brain, there is a less movement of, so this is a kymograph. So in the kymograph, the less the lower the line, that means the lower the mitochondrial movement, because x-axis is the time. So we actually saw that in low cultures or Alzheimer's disease cultures, the mitochondria are less moving. So that means there is, so what have we saw that in Alzheimer's disease culture, the mitochondria are less healthy because there are less mitochondrial protein, but there is phosphorylated tau. And now we showed that the mitochondria are not moving too. So that actually provides good support to our hypothesis. So, and here is, we are also showing it statistically that this Alzheimer's disease cultures, so they have less mitochondrial movement. So the mitochondria are not moving due to the hyperphosphorylated tau. And then we applied CP2. And we saw that in control or healthy people's brain, this CP2 or our developed drug, so they does not increase any mitochondrial motility. But in Alzheimer's disease brain, it actually increases the motility, which was decreased due to Alzheimer's disease. So that adds a new mechanism to our drug and also a new phenotype to the Alzheimer's disease field. And we also detected the length of these mitochondria from these videos. The software just gives it automatically. And we actually saw that this green one is the control, healthy control, and the red one is the Alzheimer's disease. And you can see that in the Alzheimer's disease, the mitochondria gets kind of elongated, but if you add CP2, it goes back to this black one, or it decreases again. So that's another phenotype we can get it from here. And at the end, we also did this experiment where we have a seahorse. So this is a machine. So as we are saying that mitochondria can produce energy, right? So this indirectly can kind of shows that how much energy mitochondria produces. So in simple words, so here it's showing the mitochondria's preparedness for upcoming energy stress. So how prepared is the mitochondria for upcoming energy? That means that if you were like say running a lot, right, and you are like exhausted and you are like sweating, so will the mitochondria be able to provide you more energy, or will it just get not give you energy and you will just collapse. So that property is here, you can see, was decreased in load or Alzheimer's disease neurons and astrocytes. And because this green one is the healthy controls and this red one is the Alzheimer's disease. And here we showed that if we add CP2, this drug, this preparedness actually increases. So now, we got this patient-derived Alzheimer's disease models. So now at an industry setup, my work is to use all these techniques to develop a screening or assay model to make a new Alzheimer's disease drug. Because like probably have heard that there are like some new drugs recently for Alzheimer's disease, but none of them actually works like super great. And so the Alzheimer's patients are still kind of on vitamins and placebo kind of drugs. So probably we're thinking we have some good candidates and good leads. So we'll work with these models to make a new drug for Alzheimer's disease. So yeah, so the current and future direction of my work is to work, as I said, to model juvenile epilepsy research and also collaborative research on novel Alzheimer's disease drug development. But the potentials are unlimited because like these cultures and these techniques, like from here you can get the synaptic activity, you can get mitochondrial activity, and you can use omics to get all of the proteins inside them. So we are also extending this method to use in the AIDS research, because we recently found out that the AIDS therapeutics, CAR-T, so that actually produces autisms and intelligence deficiencies. So these are the few projects I'm also working on right now. So that was the whole presentation. Sorry for boring you. So I am thankful to these two guys who taught me everything, Professor Bardishevsky, my PhD mentor, and on the right, Professor Trushina, my postdoc mentor at Mayo Clinic. And thanks for all the fun they gave me. Thanks. Any questions?
Outstanding presentation. Okay, so anyone have any question, please unmute himself or herself to ask questions.
Yeah, Mahmoud.
Okay, I have a question.
Yeah.
Yeah. So, do you think that the 3D culture means can replace the animal work?
That's the goal, actually. So one of the grant I'm writing right now is that so there are like multiple phenotypes, phenotypic outcome from a single models, right? So in animal models, you can probably say glucose, say maybe PET scan or some other phenotypes. So what people are trying to do with AI right now is that you get like multiple phenotypic readouts and you plot them together, right? So at the one spectrum of the plot, that's like the disease state. And you put lots of, with the AI and PCI clustering, what you can do is that you can put a lot of intervention or like say drugs or anything. And what happens is that the points will shift toward the healthy state if the drug actually works. So this kind of system is like people are relentlessly trying to make. And what happens is that in animal models, so at the clinical trial level, right now the efficiency or the predictability of a hit or a drug that actually passes the FDA approval is really low. It's actually 60%. And if your probability is 50%, that actually means nothing. That means it's a 50-50 chance, right? And right now it's like 60%, just 10% above the line. On the other hand, if you are using this much things like metabolism, mitochondrial tracking, synaptic, and this kind of steps. So our claim is that we can push it to 90 to 95% with the stem cell kind of related work from patients. So right now, I don't know, but that's the goal, and that's everything we're working for, to not only like replace but surplus animal models in future. But one thing I know you will say that what happens if like there is a kidney failure or a heart failure for a brain disease drug, what happens then? So yeah, that will be a moot point and a limitation of this technology.
Yeah, you're right. And some other points means in 3D culture, you can means assess the molecular effect, everything you can assess. But in the animal model, we can also assess the functional benefit, means how they act, means we have some behavioral assessment, we have some other assessments. Let's say in the 3D model, you can incorporate with these. So this is the limitation. But the good thing is it's a human model. Yeah, definitely. It's a good thing is it's a human model. So what I'm thinking that means it's not replacing the animal model. It could be supplementing or the additional, the next step. So now we are doing the bench work, then animal work, then the clinical trial. So what it could be, that's the bench work, then animal model, then the 3D model, and then go to the clinical trial.
Yeah. So that would so I had this discussion like multiple times. So that's the like there are like two kinds of people now. So I'm neither of, because I'm too small to have my own opinion yet. So my previous PI, Dr. Trushina from Mayo Clinic, so she used to think exactly the way you did. But my current CEO thinks that he can surplus it. So let's see what happens.
Okay, good luck. So I think anyone has any question, please.
Yeah, Dr. Mahmoud.
Yeah, can I?
Yeah, sure.
Hi, doctor. It's a wonderful presentation. So yeah, may I know any mathematical model behind it?
So so the mathematical models I'm not showing here. I haven't shown here yet, but like the few mathematical models, one of them is used here is this one. So here you can, there is like movement of mitochondria. This is like a simple traffic monitoring kind of thing, but it can become difficult when there is a like fusion or fission, like when one object becomes two or two object becomes one. That's one. And another one that we published in Nature is this one. Let me show this one. So here what we did is that we, with the basis of experimental data points, we made a mathematical model to create clustering of cells, different kinds of cells, neuron and astrocytes inside the confinement. So that gives us a new tool to predict how a cavity in a brain will react. So
yeah, all about, did you use any first order derivatives like something or
yeah, like there are like lots of it's a big code. Like it's like for here it's a force model. So there are like so what happened is that we have neurons, like different kinds of cells, and we measure their interconnected forces. And with that force, we like there are some one part of FEM, finite element method is also used at some points. And the other parts are just basic physical mathematics.
Yeah, you mentioned about the voltage is rising, and then it should give the message to other neurons. Yeah, that's nice.
Yeah, so that's epileptic seizure models, but I kind of left it because like I didn't have time to pursue that research any further.
Okay,
Yeah,
thank
but
you.
that was an interesting thing that we
There's
could
a
have got
huge
from
thing
this.
inside. Yeah. Thank you very much. It's a wonderful, eye-opening research. Wishing you all
Thanks,
the
Dr.
best.
Mohammed.
Yeah, thank you.
Yeah, thank you. Does anyone have any question? Okay. Sorry, Fayad, I have another simple question.
Yeah.
Okay, so you said that in your future work, you are developing a 360
86.
386 plate that you can use 380 drugs in a one. So my question is, though, from where you is it from one patient source? The model that you are
we'll just
creating?
make we'll just yeah.
Sorry.
So no. If you finish first.
Is it only one patient source? So means you took then one epilepsy juvenile patient and make that model. So it is the only one patient, isn't it?
No, so the main thing about using this IPSC model, so that's another drawback of this area is that you at least have to use 3 patients, because human to human variability is a lot. If it's not like a super bad mutation, super defined mutation, which is not the case for epilepsy. For Alzheimer's disease, like if you work with APP, pre-cell mutation, so then it's defined. But in epilepsy, it's not that simple. So you have to have a good amount of participants. So that's why doctors are super handy to collaborate with doctors, because that's why we're collaborating with this Dr. LaRocca in Stanford, because he has all these patients and he can give us these cell lines. But we'll use at least. So my right now goal is because like I'm not like little bit less into academia, and academic research is still a part of it. But the thing is that so these systems, the 386 well players and the full camera systems, so we develop these systems and all the softwares and AI-based analysis softwares behind these things. So we develop everything and we sell them. So we'll make these systems available to everyone, whoever want to give us money. They can give us money. This assay as much cell line as they want. So yeah, that's the primary and final goal.
Thank you very much. A nice presentation, of course. So now, thank you everyone. So we will have a next session in the next week, hopefully. So thank you everyone for joining us. Now I would like to finish this session. Thank you, Pratik, and thank you everyone.
Thanks.
Bye.